Hi students, today we will discuss how a blast search is performed to analyze the sequences and to confirm to get a particular sequence from a database. So starting with the topic, what we are going to do, we are going to analyze sequence using NCBI, that is NCBI BLAST. BLAST stands for Basic Local Alignment Search Tool and and now we need to know about first what is NCBI. NCBI basically National Center for Biotechnological Information. This is the website where we can get into this very important tool. And NCBI has a lots of tools available on it. It helps us performing various analysis. First, for analyzing sequence, we have to go to the nucleotide section and we can get a sequence if we want a sequence similar as sequence homologs from a particular organism. So we can go to a nucleotide, then we have to search. For example, if you want a HSP protein, HSP19 in Arabidopsis thaliana, we will get the sequences from nucleotide section. So here it will show a list of sequences. So from there we have to select a particular sequence what we are looking for. So you can see we can get a sequence from here and you have to click on the click. This will open the sequence information and where we can get this link like genomics sequences or protein sequences, a particular gene, mRNA. So we are interested in mRNA. So we will click on mRNA or RefSeq. So we will get this HSP protein sequence. HSP 90.1. We will get its gene sequence from the CDS. So a coding region that codes for a protein will be selected from here. And after that, we copy, go to the copy, and then first we have to select which type of analysis we have to do. So we are going for blast analysis using nucleotide sequence and we are looking for a nucleotide sequence. So we will use here nucleotide blast. So this will give an output in the form of nucleotide sequences against the sequence we have searched. So we have to first paste the sequence here and after paste the sequence, if we we have to get particular setting here which database we need to search. We will fix from here. Its RefSeq sequence means mRNA sequence database only will be searched because it will search in the genomic sequence, in non-coding region, in mRNA sequence, in alternative splice variants, and many other databases are embedded in the BLAST search. So we need the sequence of mRNA. So we will search against RefSeq RNA sequences. So after that, you have to go for settings like we want a sequence, similar sequence from your tomato. So organism, sorry, Solanum lycopersicum. This organism is selected, and when we go for a similarity selection or a program we need to select, we either can select highly similar sequence or somewhat similar sequence. So after selecting somewhat similar sequence, we get as result in few minutes because it takes a large data set to search for. So it gives us the result in graphical and tabular both form. Here, in this section, you can get the sequence name. You can see Solanum lycopersicum HSP90 protein mRNA, and here you is the scientific name of the organism, that is Lycopersicum, Solanum lycopersicum, because we have selected Solanum only, so it will show the result from tomato. Maximum score you can see here. This is the maximum score. What I'm clicking here. So higher the sequence, higher the similar sequence. Higher is the score, higher is the similarity with our desired sequence. Desired sequence. Then how how much your query coverage is here, that represent how much similarity is with our target sequence. Then you can see identity, how much identity that you can see. It's, if it is 100% means all both the sequences are totally same, but mostly it doesn't happen. So if similarity is very high, so you can get the similarity between two sequences higher. So that is more similar to our target sequence, and the accession number we can just click from here. You can get the sequence from here. You can just click, and a sequence what you are looking for will open in a new window. So there you can see the description of the sequence. So here you see it's the authors who has submitted these sequences to the NCBI, their description, their description and other things has been embedded here. So from here we see this gene information, it's primary information, gene and exon positions from where this is present, CDS. You can get the coding region here. You can see from a mRNA sequence the start codon and the highlighted region showing the CDS, so you can get the exact coding region sequences from here. Here, what you also can do, you can also go for graphic summary, where you can get the similarity and with the sequence how much similarity is showing sequence wise, and in the alignment you can get once your target sequence, your query sequence with a found sequence, and its pairwise alignment means between only two sequences alignment is performed, and it's shown in with the this, sections, like you can see where the gaps are there, where the similarity is here, it has been shown here. So it has been with all the sequences found by the search. So here you can perform different type of blast analysis, because once I go, when I'm showing here, the second option is blast X, where your nucleotide sequence is translated to a particular amino acid sequence, and this amino acid sequence search for a protein database. Means your nucleotide sequence just directly search for a protein sequence. T-BLAST shows your translated nucleotide sequence, sorry, a protein sequence collects information about a nucleotides sequence, which is searched through a translated way. Means protein sequence look for a translated nucleotide database. Means you get the output in the form of a nucleotide sequence. And another blast mode is protein blast, where you get when you search a protein sequence, that results comes to you in the data form of a protein sequences, where you only get the protein sequence and embedded other informations of that gene. So from here, this was the total blast search we can perform here. So thank you very much.